CFTR transporter doesn't work. You can't move these chloride ions out, right? You still recording? We're not recording anymore. Let's record some more. The chloride ions can interact with water, right? Because they're negatively charged. And they help keep this water layer here on the surface of the cells. If you can't move chloride ions out here, you have less water on the surface of your cells, and what happens is you get more mucus. And the more mucus accumulates, and that tends to get bacteria, and tends to make people sick. So this mucus layer gets really thick. It just changes the ions inside the surface of the lungs, because that chloride can't be pumped out. It just changes everything. All right, and so this water that's supposed to be there, it just gets, the balance gets messed up. And I got a little video I want to show you guys, so just hang on to your questions. And let's watch the video if it plays. But you know, we're having that kind of day. Good.
A patient with cystic fibrosis is susceptible to infections of the lungs. This is due to a mutation that affects a protein on the surface of lung cells. Many different mutations can cause cystic fibrosis, but all of them involve a transmembrane protein called CFTR that functions as a channel for chloride to move in and out of cells. In this case, CFTR is less able to transfer chloride ions out of the cell compared to the functioning protein in people without cystic fibrosis. The channel is, in effect, locked shut. This inability to transfer chloride ions has an effect on the outside of the cell. In people without cystic fibrosis, there is a balance of chloride ions inside and outside lung cells, which maintains the right amount of water and salt ions in the mucus lining the airways. Cilia normally sweep the watery mucus from the lung lining, keeping the surface clean. In patients with cystic fibrosis, chloride ions concentrate inside cells and draw in water by osmosis. With less water, the mucus becomes thicker and cilia can't move. Flow is reduced, providing an environment for bacterial infection. But there are now drugs that can help patients. To find a drug, researchers tested over 200,000 small molecules to see if any would bind to the CFTR protein. Then, for each small molecule that had some binding ability, researchers chemically synthesized different variations of that molecule and tested them for the ability to improve the function of some types of mutant CFTR channels. They identified a drug that, when taken by patients with a certain type of CFTR mutation, binds to the CFTR protein, opening the channel and allowing improved ion transfer. This increases chloride ion flow and maintains a watery, clear mucus, which helps the cilia move more freely and reduces the risk of bacterial infection.
Nice. So that kind of gives you a picture of what's going on inside the lung as to why that would cause such a problem and cause those infections. Okay, so what was the question? Yeah.
So on the second part of this picture is not showing up.
Okay, I can make myself a note to come back and add that.
Okay. And then is there a link to that on the
I can put it there. Let me make myself a note.
Okay.
Let's see, pictures and video. Got it. Other questions?
You could put it in the animations. I thought it would be like.
Yeah, I can put it in there.
I saw something, it was basically like CF patients can be around other CF patients because of the bacteria or something.
So I don't know that for sure, but I mean, obviously, if you are a person who is susceptible to infections, being around other people who have infections, you'd be exposed to those infections, right? And so you don't want to be exposed to those infections. Because there are opportunistic bacteria that can infect people who wouldn't normally infect people who don't have cystic fibrosis, right? But would infect someone who does. So you wouldn't want to be around other people who have an infection if they have cystic fibrosis, because that bacteria that's in them might not affect aren't affected, but if you have cystic fibrosis, it could make you really sick. Okay. All right. So this gene has been studied a lot because it causes a pretty common hereditary disease. And there's over 1,700 mutations in CFTR that are known. So pretty hard to cure a disease with that many mutations. So there's 2 drugs that have been pretty successful, and the video talked a little bit about how they designed those drugs. One's called ivacaftor, and the other's called lumacaftor. And there's 4 specific mutations because they were designed to specifically treat changes in the structure of the channel, right? Our structure of our protein is important, right? And these drugs were designed to treat those specific mutations. Right, so this one, right, we're not memorizing this, we're thinking about the principle, right? The principle that the structure of the protein matters. This drug is for this specific variant that makes up about 5% of cystic fibrosis patients. And it is able to increase the transport of chloride ions out of the cell. So it's been pretty successful. This other drug is for this other mutation that actually is about 70% of people who are affected with CFTR. It doesn't increase chloride ion transport, but it helps the mutated protein fold. All right, remember how we talked about how the primary structure is what determines how the protein folds? The mutation changes the primary structure, so it changes how it folds. So this drug gets in there when the protein is being synthesized and helps it fold better so that it can have some function. All right, so this drug helps the protein fold properly. All right, so that's a little bit about a specific example of a transporter and why transporters are important.